The other thing which I didn't just put tonight, but hey, I just put our review right up here. I don't have to really talk about it much. Again, the metabolism chapter overall that set us up for the hormonal and cortisol was sort of this here. And again, all this was from that was pretty much this, right? The different sort of hormones and things that are controlling hunger. Okay, low blood glucose and glucagon are going to obviously that's sugar. Low sugar is going to trigger hunger. There's neuropeptide Y when you go back. It's basically remember the hypothalamus is the central guy just controlling this. And the other thing to try to get us through the digestive tract. Empty stomach produces ghrelin, that's all hunger. Satiation, when you're in the sped state. So if you have high glucose and therefore high insulin, that's going to decrease your hunger, satiation. Fat, you've got a lot of energy, produces leptin. That inhibits NPY, so that's going to be satiating. When food enters the small intestine, we want to slow down. Releases a couple hormones. The only one on the test is CCK, and that's going to basically be satiation. That. The other things, just simply because that's why we want the lead in for cortisol, you do have to remember all this. So, when you do have high blood glucose and insulin, it does these 5 things. It gets glucose into the cells and helps break it down so you can get energy within the cells. But if you're in the building type of mood because you've got energy, protein synthesis, lipogenesis, and making glycogen, right? Glycogenesis. You go to the opposite side, low blood sugar, alpha islets produce glucagon. It's going to cause the lysis of those. Break down glycogen, get glucose in the blood. Break down fats and proteins. Other tissues can use that for energy. Plus, we can use some of these products now to make glucose, new glucose, gluconeogenesis, and also turn some of the fatty acids into ketone, emergency fuel for the brain. All that's involved in glucagon, so you have to know that. All right, the 2 respiratory. So again, remember, I don't have time for all of that. I skipped all of that. You don't have the gas laws on this thing. We already took care of that. That's on the lab test. So all we have okay, so it's part of this. Okay, so and really it's going to be that last couple slides. So you do have to know this slide right here. So for ventilation, the idea of now we're controlling airflow into lungs, and so we're talking again bronchoconstriction versus bronchodilation. So, as it says up here, it's on this one right up at this point right here. You have this idea that in this case, there's sympathetic and normally sympathetic and para, okay? They're in opposition, which they are, but the bronchioles actually do not have sympathetic, but they do have parasympathetic, and that's going to cause constriction. If you're at rest, you don't need as much airflow. So you basically, again, but you need to remember the details of that. So bronchoconstriction increase parasympathetic, which of course is an increase in acetylcholine binding to muscarinic receptors. So you do know those details, because I could give you a muscarinic receptor antagonist, agonist. You've got to see how that's going to affect now bronchoconstriction or dilation. So you got to remember those details. And the other thing for that is sort of allergic reaction, histamine. Histamine binds to these histamine receptors. Okay, so that's bronchoconstriction. For bronchodilation, again, you don't have the sympathetic, but you do have epinephrine. You do have the hormonal. So increase in epinephrine. Okay, and what receptors do epinephrine bind to? Beta 2. And remember, it causes vasodilation. On bronchioles, they're going to cause bronchodilation. So you got to know that because I can have a beta 2 antagonist agonist, just have you know that. So you got to know that. So the other thing on this chapter, remember, lung volumes are all today, is this. And again, I don't like charts. We're going to kind of do this. So I want to explain. The whole idea is, let me just put a picture and then try to again make it sort of easy. Is, this is the idea of, ventilation. Remember, we're going to add, via the bronchioles, we're controlling ventilation that's coming into the air sacs, the alveoli, but also then perfusion of blood, how much of that is entering the blood. And the key is, we're going to control this. And the bottom line is, wherever there's more oxygen, that's coming in, okay, or which one has more oxygen? Because the idea is sort of to pick it up, and I'm going to draw my 3 boxes. If there is more oxygen in this is going to dilate to pick it up, okay? Our goal is to pick up as much oxygen as you can. So, again, you can memorize that chart, okay, for some reason, but the idea is, again, the summary I'm going to do is those 3 diagrams, and I'm going to have, again, the bronchioles, okay? Over here, I can use a whole board for this then, I'm going to have what we had before, which are the arterioles. I'm going to put systemic, leading into the tissue. That's from the cardiac section, but we're revisiting it. And again, we'll do high metabolism, and it relates to the gases. So high metabolism, okay, we're using up oxygen, basically having a lot of CO2. So again, if you basically have a system in which this has low O2, okay, we have a system that this has low oxygen, what is this going to do? Is it going to dilate or constrict? Well, what is this delivering? This is systemic arterial. This is back. It's going to dilate. Okay. This was called. We'll have we're going to cause vasodilation because the job of the systemic arterioles is to deliver oxygen to tissues that need it. Serve the needs of the tissues, okay? All right, so we have that. Okay, reminding us it's called active pulmonary. So think about the bronchioles, okay? If the bronchioles, we're going to keep that low oxygen right now. If the bronchioles have low O2, okay, leading to this, okay, what do you think the bronchioles are going to do? Are they going to constrict or dilate? They're going to dilate because their job, again, is to deliver, okay? Just like this one. They need, their job, okay, so for low O2, it is going to cause bronchodilation. Because the job of these bronchioles are, again, serving the needs of the alveoli. They're going to deliver O2 to the alveoli. The idea is if you don't have enough, of course they're going to dilate to get more. Okay? If you don't have enough, these are going to dilate to get more because their jobs are delivery jobs. But now we're going to look at a situation like this. Okay? And now we're looking at, remember, we have millions and millions of these alveoli. Drawing these, but we'll put this. Okay, so they got a different color. These represent the alveoli. But now we're going to look, so I did the opposite for color-wise, but we're going to look at this idea, of course, we have all of these, okay, these pulmonary arterioles. So not the systemic ones, the ones that are basically flowing through. And in this case, again, if this has low oxygen, so we have the relative difference of oxygen on these alveoli, and we've done, again, this had low oxygen, this was going to dilate to deliver oxygen. If the alveoli have low oxygen, the bronchioles are going to dilate to basically deliver more oxygen. If we have low oxygen here, the pulmonary arterioles are going to, this is going to cause constriction. This is going to cause dilation, because the job of these, just to make it a job of the pulmonary, is to basically pick up O2 from the lungs. They're not delivering, they are picking it up to get it to the body. So again, they've got a choice. You only got a small time to do it. You're getting paid by how much you get. Okay, you got a place which has low, a place which has high. Where are you going? Well, a place that has high. So it's the opposite, and that's why, again, they sort of show that. Okay, again, you had low O2 on the right. Hey, it says, forget it, I'm going to that side. And that's where, I'm not going to use that particular term, but it actually is. The idea of this is ventilation, how much gets in, is going to match how much gets in the blood. If more gets in, these dilate you to get more in the blood. The job of these guys are to pick up, not deliver. And that's why when you look at that chart, they're the opposite of what these guys are. That was a hard concept from that chapter. Okay, so you memorize that for some reason, or try to understand it from this and remind yourselves. Last couple things, chapter 18. So two things on this chapter that are the big guys. One's bigger than the other. So all that you have to really know. So you've got to know these different types. And then we go hemoglobin saturation curves, and then we're done. So 4 types, again, well, you got to know the term too. I mean, I think I used hypercapnia on the test. Remember, hypercapnia is an elevated amount of CO2. But here, we have the alveoli. Okay, here's the blood, the plasma of the blood with our red blood cell, and of course, our hemoglobin. Okay. Here is our heart. Here's our cells / tissues. O2 is trying to get from here to binding. It's got to make it via circulation to the tissues, and then it has to get into the cells so we can actually use the oxygen. All right, and that's where that kind of comes in, because there's sort of 4 types of hypoxia. Bottom line is, if these cells are not getting enough usable oxygen, it's hypoxia, right? They're not getting, that's what hypoxia is, right? But you can have different ideas. So the first one, if you're not getting enough air, basically, enough oxygen, from, into your blood from your lungs, that's going to be toxic hypoxia, okay? So 1 is that first one there. You're not able to get it to this point right here, okay? Again, lots of reason. You might have COPD where this is very low. You might have fluid buildup or anything like that. Anything that's causing this to be compromised. You're not getting in here the toxic hypoxia. Second type of hypoxia is anemic. That should sort of make sense. If this is the problem, so that you don't have enough functional hemoglobin, not enough red blood cells, or this doesn't work, the idea is, hey, you're not going to get it to the cells. So that's anemic hypoxia. 3, if all that's working and you're loading up, but your circulatory system is compromised, You're not going to get enough to the tissues. That's ischemia, right? That's ischemic hypoxia. That's 3. And the fourth one is if you have some sort of basically poisons, and you can see cyanide is one of them, that basically don't allow oxygen to get in the cells and be usable, to actually be used, that's, toxic. So it's toxic hypoxia. That's why I like drawing the diagram. Okay? Final thing for us to review is hemoglobin saturation curves and the tips. So we'll put them up and see, at least you'll have this to look at. Remember, of course, you're not drawing this, luckily. 0, 40, 100. So remember, but you'll see this. The y-axis is basically, and we'll kind of remind ourselves one more time, hemoglobin, which can bind 4 molecules of oxygen. Again, 2 alpha, 2 beta on adult, but we'll get this. This is the percentage of saturation, saying that if all the hemoglobin had 4 oxygens, it would be 100% saturated. So 5, 50, 25. And this is the concentration of partial pressure of oxygen that is surrounding this, anywhere between 0 and 100 millimeters of mercury of oxygen pressure. Okay? But again, and let me try to draw something like that. All right, so first thing we have to know is what's happening, basically, or what these curves are representing for us biologically. And the key with that is, again, 100 millimeters of mercury is where, when you go back to this, here's the alveoli, that's when the hemoglobin is being exposed at the lung, which is 100. And what you see is basically that, and that's why actually we'll just do it this way, like this before. Brushing a little too much here. So we want to do Stu's way of reading in Hebrew style starting over here, and you can see basically on that too, on this slide right here. This part of the curve is again loading, and it looks like shape is a plateau. Okay? So when you're looking at this, again, I don't know if necessarily want you to mess with that because it's in here too. If you look at this first, or if I'm asking you what's going on here, which is obviously what I'm doing, what I could be asking is, this is where it's loading up. Hemoglobin's becoming oxyhemoglobin, loading up. And you can see it's at the lungs, which is when it's 100, it basically is almost 100% saturated. That's how efficient it is. It sucks it up. Okay? That's why it's a loading phase. It's when it's loading. The idea of a plateau, as far as significance to us, is what if your alveoli pressure is compromised by all these things we've talked about, where you're not getting enough in? Well, instead of being down here, the hemoglobin still is almost 100% saturated. So if it's less than optimal, you still can get into your body pretty efficiently. And that's the advantage of it being a plateau. So it's loading, and it's basically protecting us if we don't have optimal conditions. And that's what you need to know about that. If you go to what you need to know about the steep part, it's written on there as well. But on the steep part, we're going to call it the unloading, okay? And basically, the idea that it's a steep curve. It's unloading because it's the idea of that concentration, when you go back to 40, is basically the concentration that these hemoglobins and the blood cells are encountering at basically relatively metabolically inactive or resting cells. Okay, they're obviously working, but that's about that. So you don't want this to hang on, you want it to deliver, and it doesn't say, it actually does. So it's unloading some of its oxygen to those cells because they need it. It's not 100% saturated. So if you kind of look at saturation on that, you see kind of at that, it's still 75% saturated. So in a way, you can think of it for us, you kind of released around 25%, okay, for a resting cell. Okay, we don't want it to give it all up because what if you have a metabolic active cell that needs more oxygen? So the more active it is, remember we just had that up there, the less oxygen, we want it to deliver more. So the advantage or the significance of it being steep is it's going to be responsive to the needs of the tissue. if it needs more oxygen, less saturated, it delivers more oxygen. The more it needs, basically, the more it's delivering. If it stayed up here, it'd be ignoring that. So that's the idea of the steepness. It's responsive to the needs of the tissue. So the last thing to do are these shifts. And all you have to remember, again, at least I think to remember the shifts are, because you're going to see to the right and left, a shift to the right means basically the oxygen has, we'll put it in those terms, less affinity for hemoglobin. But basically for us means hemoglobin is releasing more O2 to the cells. Okay? That's going to be the significance of this. Whenever you need more oxygen, or think that would make sense, you're going to end up seeing a shift to the right compared to the left. Okay? That's going to be the key for these curves. And so to relate most of them, except for the fetal one, the idea could be you could basically think of, and we'll do this, high metabolism. Which again, that means low O2, high CO2. So think of high metabolism. Also, let's remind us of CO2 plus H2O, just to remind us that CO2 is related to pH. Or this is pH. Okay, and just remember, so just the basic bottom line, if you have things which are associated with high metabolism, do you think your cells need more oxygen? So you're going to see a shift to the right. Okay? That's all you have to remember, and then this should make more sense. So obviously, under these conditions, you see, anytime you need more oxygen, you're going to see that we have it where certain things happen to this, where it releases more. And so, basically, you could think of temperature, okay? So, high metabolism, what's the temperature doing? Up or down? So, you should see a shift of this curve to the right. Okay. What is CO2 doing? And so you're having high CO2. So for CO2, you should also see a shift to the. As CO2 is going up, H plus is going up. We're used to that. pH is going. So low pH, you should see a shift to the. And you do. Here's my little, right, I can do this. If you go, let's see in a second. We'll do it in order. So you see, of course, we won't get up to 43, I hope, but the idea is higher temperature, shift to the right. Again, you can go back and realize this when we see more, but it's consistent. High temperature, shift to the right. High CO2, shift to the right. Low pH, shift to the right. So you can memorize any way you want and all that, but for me, this makes the most sense and easiest to memorize and actually has some functional significance to it. Okay? Again, the way this is happening, temperature is changing, hemoglobin changes shape, it releases more. You have high CO2, even though I didn't talk about it, hemoglobin also basically carries CO2 in our body, but binds to CO2, changes the shape, releases more oxygen. This also binds H+, which is acid. The idea is, therefore, when you have low pH, a lot of acid, it changes the shape of it, releases more oxygen. But the idea is, all those conditions can shift to the right. So, hypercapnia, metabolic acidosis, hyperthermia. I don't have to do exercise, right? Anything increasing the temperature, CO2, lowering the pH, which you can always remember by that, is going to shift to the right. If you had hypothermia, okay, left. Okay, am I going to have some of these left? High pH, yeah, it's the opposite way. Am I going to have some? Yes. So you're going to have a picture. I have some questions about this, and then I'm going to have conditions, and really, you can, again, it's going to be shift to the right, shift to the left, doesn't do anything. I don't think anyone's gonna put that. I don't even know if I have a D. All of the above. I mean, what can I do? It's really shifting the right or shifting the left for these. Okay? 2 more curves, and then we are done. The other aspect is this. And again, you're trying to look at this. If you have conditions in which you are not getting enough oxygen, you have to use anaerobic respiration more. That's glycolysis. And so one of the intermediates of glycolysis is this molecule called 2,3-DPG. It does double duty. And the bottom line is too, if you're not getting enough oxygen, you're in an environment not getting enough oxygen, do you want hemoglobin to deliver more oxygen? You're not getting enough, what do you want hemoglobin to do? Keep it or give it to you? And so you're going to shift to the right. Okay? Any condition in which you know you need more oxygen, it's made to the shift to the right. So in this case, it has another site to bind 2,3-DPG. So it goes over and binds and basically, again, causes it to shift to the right. Okay? Those are 4 of the 5 curves. We've talked about shifting to the right. Here it is. The last one, and the last thing I got to talk about, which is good, is carbon monoxide, fetal hemoglobin. So fetal hemoglobin, I'm going to just write this too. So maternal or adult, R's, versus fetal. So fetal hemoglobin, we're going to see a shift to the left because the conditions of the fetus are not basically exposed to those concentrations, and if you kind of just examine which we're not, this is going to more fit, the concentration it's just being exposed to pick up, and sort of its oxygen level within its tissues, it's going to match. So you shift to the left is what you need to know. The other thing you need to know, and I'm done, is sort of why that hemoglobin actually shows that pattern. Is because maternal or adult basic hemoglobin, when you look at it, has 2 alpha and 2 beta chains to do its function. The fetal, 2 alpha, and the other ones are gamma chains, which give it slightly different properties, and basically those properties at all. So different hemoglobin because of these gamma chains. Again, once the baby is born, it's got to get rid of all that. That's why you potentially can be jaundiced and replace with this. Okay, so that's